This lecture will be about bootstrapping, which we can use to have some estimates on confidence intervals or uncertainty in parameters or other conclusions that we take when we adjust some model or compute some results, for example, like phylogenetic trees. So I'm going to talk very briefly about resampling methods and what they are used for, and then we're going to focus on bootstrapping and look at the variant also residual bootstrapping, which we can use when we cannot really sample our data at random because some of the values are chosen by us. And then we're going to see an application of this, continuing what we saw last week in the calculation of phylogenetic trees from multiple sequence alignments, and we're going to use bootstrapping to evaluate the confidence we can have on each branch of the tree. So let's start by looking at resampling methods. The idea here is that we have fit some model. We computed the best parameters to adjust to the data. For example, looking at the example from the previous lecture, we see that we have this curve for the Michaelis-Menten kinetics, computing these parameters for Vmax and Km. And this is the curve that best adjusts the data by minimizing the sum of the quadratic error. But the question now is, how reliable is this? What is the uncertainty we have on these parameters? And in theory, what we mean would be, how different would this be if we were to repeat the experiment? So basically, if we could do the experiment again and again, we would obtain slightly different values for the initial velocity measurements at these concentration values, and this would give us different results. Using only the data that we have to fit the model, we cannot really estimate how good this would generalize for different experiments, for repeating the experiments or applying this to other cases. So this would be a general question. Once we fit a model to the sample of data that we have, how do we know if it works well if we apply it to a different sample taken from the same population? So repeating experiments with the same reagents, or taking a sample from, again, from some group that we are observing, and so on. So one answer for this is resampling. Basically, we are using the sample that we have, the data that we gathered, and use that to obtain different samples in order to evaluate the fit. These first we're going to see in more detail in a few weeks, which these are applied mostly when our concern is whether or not the fitting that we did, so the parameters we computed for the model, give us something that we can generalize and apply outside our data. So this could be splitting our data into a training set and a test set, so that we fit the model to only this part of the data, the training set, and then check what happens outside the training set, in the test set. This would be the train test split approach. A bit more sophisticated is to split our data in a number of folds, k folds, and then we're going to fit our model k times, and each time we leave one fold out for validation, for checking how the result performs outside of the training set. So these are widely used if our main concern is generalization. After fitting our model, the result that we have, the trained model, how well does it perform outside the training data? But for now, in this lecture, we're going to look at a different resampling method, bootstrapping, that can also be used for this, but it's often used to estimate confidence in the parameters. And this is, bootstrapping involves obtaining new data sets by resampling from the sample we already have, instead of repeating the experiment or obtaining new data from reality. So that would be ideal, but in general we cannot do that. So instead of obtaining a new sample from the source, for example, by repeating experiments, we can resample the data that we have and create replicas of our data that are slightly different sets, but have the same distribution as our own sample. So let's look at this in a bit more detail. This is bootstrapping is a method of generating replicas of our data set by sampling from the data that we have. So we have one set of data with n examples, n points, and now we can generate a set of replicas by drawing at random from the sample that we have, from our training data, the same number of points for each replica. So basically, since we are drawing with replacement, this means that we can draw some examples more than one time. So we can have duplicates or triplicates and so on in our replicas. And doing this sampling at random with the uniform distribution and replacement, on average, each replica will have 2/3 of the original examples in the original data, and then the remaining third will be duplicates. So the same points that we have on those 2/3, some of them duplicated in this third part. So this means that each replica will have a different set of points than the original, and also differences between the replicas. So now we can use these replicas to fit our models and then take some estimates on statistics, like the distribution of the values for our parameters and so on. Let's see a very simple example here. Suppose that we want to compute the average height, the mean height of a group of people, for example, students in some college, and we have a sample. This is our sample of students, the height in meters that we have. We could just take the mean of this sample, and this would give us an estimate of the mean of the students on this campus, but it would not give us an idea of the uncertainty that we can have on this estimate. So we can use bootstrapping for that. We're going to create an empty list here and then repeat 500 times. By the way, this underscore is a convention. Underscore is a valid variable name in Python because you can start variable names with underscore. But if you call a variable just underscore, you're telling the editor, Spider, that you don't care about that variable. So it will not complain if you don't use it later. And this is an example where you may want to do that, because the for loop has this syntax. It requires a variable to point to each object in the iterable that you use here. But we just want this to repeat this code 500 times. So we don't really care what the value of this variable is at each iteration. And so instead of calling it, for example, I or j or something like that, which would lead to a warning because we don't use this variable, we can just call it underscore, and this means we don't care about the variable. It's only there because it's required by the syntax of the for loop. So now what we're going to do is we're going to generate a list of an array of random numbers that range from 0 to the length of the sample, to the number of elements we have here. It will this randint function will never return a value that is at the maximum. It will always end before the maximum value, and the size is the number of elements we want in this array. So, for example, if we run this like this, we want a set of random integers from 0 to the length of the sample and an array with this size, we get this array here. So this gives us a random set of indexes for this sample that we have here. Each time we run this function, it returns a different set. So this is why we're going to use these random functions, so that we can call them over and over again, and we get a different set of indexes, so a different replica of this sample that we have here. So we obtain these indexes, we index the sample using these random indexes, and we obtain, for example, in this case, it would be the element of index 7, 6, 7 again, 1, 6, 6. So you see, in this case, by chance, 6 happened to appear 3 times, then 3, 8, 4, and so forth. So some elements are missing, others are repeated, 6 and 7, and this would give us a slightly different mean when we compute here the mean of the resulting array. And now we're going to append this to this list. So this list will give us 500 means, each computed from a replica obtained by bootstrapping from the original sample that we had. So now we can compare these results. This would be the mean of the sample. The mean of the means of all the replicas we can see is very similar to the mean of the sample. This is intuitively obvious because we're just drawing at random from there a large number of times and then taking the mean of all the means. But more interesting is the standard deviation of these means. So this is how spread out about 2 centimeters and something, the standard deviation, gives us an idea of the uncertainty we can have on this computation, assuming that this is representative of the population and that we are getting replicas that are also representative. So this is the part that we would not be able to estimate without bootstrapping, because we would only get the mean and not any notion of how the mean can vary if we draw another sample and another sample of subjects from this population. So this at least gives us an estimate of that. So this method is useful when you can choose elements at random. Sometimes you cannot simply choose elements at random. So we're going to also look at a variant of bootstrapping, which is residual bootstrapping, where we resample the error that we have instead of resampling the actual examples, the actual points in our data set. So one situation where this can be useful is in the Michaelis-Menten model that we saw previously, where we have these S values, the substrate concentration values, are chosen by us during the experiment. So it would not make sense to choose a subset of these points at random, because we would never do an experiment like this where we would do twice at this substrate concentration and then not do at this one, or omit some value here and repeat some other, and so on. So that would not make sense, because the values we have here on the substrate concentration are chosen by us to try to get a good curve here. So we must retain these values constant. And what we're going to resample is just the residuals, which are the difference between the best curve that we found here, our original fit, and each of the data points. The idea is that although these values are chosen by us, so they are not random, we are not going to resample them, the error that we get on each of the points is at random, and it's drawn from the same distribution. This is what we are assuming here. So if the error is drawn from the same distribution, we can create a replica of this data by resampling this error and assigning it at random to different points. So we're going to use this to create different sets of data by scrambling the error, by selecting at random with reposition all of those residuals, and moving the points to the position obtained by adding to the original prediction these new randomly sampled residuals. So now for each of these data sets, we can compute a new curve, because we can fit the parameters of our Michaelis-Menten model into these new sets of points. And so we have a family of curves. Each curve will have its own Vmax and Km parameters. So now we can get an estimate on the intervals and the distribution of values for these parameters over this large number of samples. So this is how we can do it. We can create this function where we're going to do residual bootstrapping over n replicas. And this is the original parameters of our curve. So we did this original fit. We have the Vmax and Km parameters. We have the original data. And now we're going to create replicas by residual bootstrapping. So first, we're going to compute the predictions at the concentration, the substrate concentration points corresponding to our data. So the predictions will be Vmax multiplied by each substrate concentration value divided by Km added to each of the substrate concentration values. So this is the equivalent of computing the predicted values at these points here with the same concentration, substrate concentration of our data points. So now we can compute the residuals by subtracting from the data our predictions. And this gives us the difference between the real values and what our model predicted. So remember that the second line on our data has the initial velocities, which is what we're trying to predict here. Okay, so now we can create a list where we're going to store our parameters. We're going to iterate through this number of replicas. Since we don't use this variable, you can, if you want to avoid the warning there that we're not going to use the variable, you can name it underscore or whatever you want. But remember that if you don't name underscore, you'll see probably a yellow sign there indicating that you're not using this variable. But this loop is only used to repeat this for n replicas. And now we're going to create a set of indexes that goes from 0 to the number of points that we have. Actually, this first argument is optional, so this also works like this. But if you do it like this, you need to specify that this is the size, because otherwise the randint will think that this is the lower bound and this one is the higher bound. So either way, you can supply these arguments as you wish, but you need to make it clear what each value is. So actually they are the same because we want to go from 0 to the number of points that we have in our data, and we want as many random integers as the number of points that we have in our data. So these will now be the indexes of the residuals that we're going to select and create a replica. So the values of the initial velocity in our replica will be the predicted values plus a random sample with representation of the residuals. So we are going to pick these residuals at random, add them to the predictions, and use that to create new sets of data. And now we're going to fit our model to these points. So using the replica, using the substrate concentrations in the original experiment, but now the velocities, the initial velocities in this replica obtained by residual bootstrapping, and we're going to append the result. So the Vmax and Km tuple here, we're going to append it to this list and return this as a matrix. So now we have on each row of this matrix one of the n pairs of Vmax Km possible, obtained by fitting the curves to the different data sets. So we can compute the mean, and this tells us the mean of the Vmax and the Km values that we have here. But we can also check, get the standard deviation to get an idea of how spread out these values are. So you see that Vmax is 73, but we have a variation of 4.2 about that. We can also, this is an idea of the different curves that are generated from these replicas, but we can also take those values and create histograms, for example, to check the distribution. This would be the distribution of values for the Vmax parameter. There is a peak here around 74, but you see that there are many cases with lower and higher values, ranging from 60 to 85. And the same thing for Km here. So using residual bootstrapping in this case, we can get an idea of how we can, of the distribution of the parameters that we found. By the way, we can do this. This is the same library that we saw in the previous lecture, pyplot, but you can use hist in pyplot, giving a set of values. So in this case, it's the first column of that matrix. You can set the color for the bars and the line width and so on for these black bars here. You can also specify the number of bins for the histogram. If you omit it, it will be computed automatically. But this is a very practical way of looking at the distribution. You just give the set of parameters. If you don't want to change the color or anything in the display, then you can just give the set of values and get a default histogram. So using pyplot just to look at the data is very easy, and you can do it like this. These figures here are an instruction for pyplot to create a new chart. So if we omitted this one, we would get the two histograms drawn on the same chart. So this way we create a new chart and then draw the second histogram. By the way, the colors can be specified by name. For example, you can say red or something like this, or you can specify by red, green, and blue as a tuple. So in this case, this orange is full red, red of 1, half of green, and 1/3 blue. And this gives you this color. This one here has 70% red and green and 100% blue, so it has this grayish blue color. Okay, but this is just for visualization if you want to see the data. If you want more details on plotting, you can check the documentation on matplotlib on pyplot, and you can see lots of different examples. Okay, so now let's do a more bioinformatics application of this, and a bit more extensive, to evaluate those phylogenetic trees that we computed last week. So we previously created this tree by computing the multiple sequence alignment and then using UPGMA on the differences between the sequences in the multiple sequence alignment in order to compute this tree relating different mammal species. The question now is, how reliable is this? Can we have the same confidence on all of these branches? And we can estimate the reliability of different branches in many different ways. There are methods for determining consensus trees, sophisticated bootstrapping methods, and so on. We're going to ignore all these complexities. So we're going to do something that is very basic. It's not meant to be a practical application. If you actually want to work with phylogenetic trees, it's best to use more complex algorithms. But our focus is on the basic idea of the algorithms and data structures. So what we're going to do is we're going to compute the tree from the multiple sequence alignment. And then we're going to create replicas of the multiple sequence alignment by bootstrapping on the columns. So the idea is that we are going to select with reposition a set of columns from our multiple sequence alignment. Remember that each column represents the residues that are a match on all of the sequences. And we're going to create new multiple sequence alignments that have the same dimension as the original one, but consist of a random sample of columns from the original alignment. And this is bootstrapping. Now, for each replica, we're going to create a new tree, and now we're going to check each branch of the original tree. So each node that we have here where we branch and have a subset of species, we're going to check if that one exists in the replica. So basically, we're going to check for this branch, we're going to check each replica tree if these two species are grouped together in a single branch like this. For this branch, for example, we're going to check if these three species belong to a clade, to a branch of the tree, and so on. And this counting how many times each branching appears in the replicas will give us a measure of the support that we have for each branch in our original tree. Now, we're going to reuse previous code from the multiple sequence alignment. I'm going to assume that this code is saved in a file called msa.py, but if you chose a different name for the file, you can use a different name in the import. But we're going to be reusing the code from last week. However, we need to make some changes because last week we did not really need to create an explicit tree. So if you recall, we used lists of sequences for the alignment, lists of strings for the names, and then we just followed the UPGMA algorithm step by step using the matrix of distances to identify which elements to merge together. These elements can be individual sequences or groups of sequences, and then we use that to build the NAWIC file, or the NAWIC string, the NAWIC text specifying the tree. So basically, we have a list of strings with the NAWIC entries, and as we combine elements from the original multiple sequence alignment, we also combine strings from these parts of the NAWIC representation until we obtain a final result which has only one entry for the NAWIC string, and we return that particular string. So previously we did not really need to create a tree in any specific data structure because we only needed to follow the correct order in UPGMA to create the NAWIC text. Now it's going to be different because since we need to compare different trees, we're going to actually create the tree because we need to check for each replica which branches in the original tree are present. So to do that, we're going to create this data structure. We're going to create a tree by chaining together dictionaries, and we're going to create in each dictionary these four keys. So these four entries in the dictionary. The total length will be the total branch length so far, counting from the end. So, for example, the terminal nodes, these leaves, will have a total length of 0. They are at the end. Then when we reach this part of the tree, we have a total length of 0.5. In this case, remember that the difference between the two was 1, so we split 1/2 on each side. And then when we reach here, we have the length that goes all the way down to these leaves, and so on. So this would be the total length that we're going to need to create the branches, the length of the branches when we create the NAWIC text. Support will start at 0, and then we're going to count for each branch in our tree how many times those elements appear grouped together, and each time we find this in a replica, we increment the counter for support. So if support is equal to the number of replicas, this means we found that particular grouping on all trees in all the replicas. If it's smaller than that, then it means we only found it in a few cases. Elements will be a sorted list with the indexes of all the elements in the current branch. So, for example, we have this branch here. These species are all present here, and each of these has an index in the list of names. So we're going to store on this node a sorted list of all these indexes. And the reason for this is that if we now need to find in a tree like this, if there is a group with all of these elements, regardless of the ordering they have, we can just compare this sorted list of indexes with the sorted list of indexes we have in that node. So this would be a practical way of determining if we have that group of species in this branch. And then we need to build a tree. So each node of our tree will also have two subtrees. We're going to call it left and right, but these are also dictionaries of the same kind, which continue the tree until eventually we reach the end, and we can just set these entries to none, because there is no more subtree. Okay, so let's see function by function how we're going to implement this. First, we need to create our replica multi-sequence alignment. So I'm going to import NumPy as usual, for statistics and so on, for arrays, etc. But I'm going to import this MSA module, which is the module from last week where we created the multiple sequence alignment. If you did not call yours msa.py, you can do something, your module here. Remember that your model is .py, but we don't put that when we import, and you can put it as MSA, for example. So this imports your module regardless of the name that you gave it, but gives it this nickname, so it will work as if it was called MSA in the rest of the code. So adapt this as needed for your particular code, but this is the general idea. And now we need to create, by bootstrapping, replicas of a multiple sequence alignment. Remember that the data structure we used for the multiple sequence alignment is a list of strings, and this is what we receive here, a list of strings. We're going to count how many columns we have in this alignment, so we can just look at the length of any of the strings on this multiple sequence alignment, because in a multiple sequence alignment, all of the strings will have the same length. They will all be padded by gap characters, so that the multiple sequence alignment is a rectangle with all those strings. So we can look at the first string, check the length of that string. This is the number of columns. And now we ask for random numbers, random integers from 0 to this value, and as many as the number of columns we have in the multiple sequence alignment. So this is basically the same recipe we've been seeing so far. Now we're going to create a new multiple sequence alignment, starting with an empty list. And for each sequence in the original alignment, we're going to create a new sequence by concatenating it with the characters that come in these random indexes. So we're going to loop through all of these random indexes, get each corresponding character in the original sequence, and append that, or concatenate that, add that to the end of the sequence. So remember this plus equal operator in Python, it means the same as doing this plus the new value. So whenever you have a variable here and then you want to add something to the same variable, you can simplify this expression by putting plus equal there. Now we have the new sequence. The new sequence is generated by this random sampling of the old sequence. But remember that we run the randint function only once for all the sequence. Otherwise, if we put this inside the loop, we would scramble everything and the multiple sequence alignment would be wrong. So we are selecting the columns, but we are not changing how the sequences are aligned. And now we return this new list, and this is the new multiple sequence alignment obtained from the original one by bootstrapping. Now we need to create the tree. So we're going to follow the same procedure as before. We're going to use UPGMA to choose the closest branches, and then we're going to join them in a single branch. So joining them will be creating a new dictionary with the two branches to join together as the left and right sub-branches, and then putting that in the tree. We're going to start with all our different species as single nodes in separate trees. Remember that UPGMA starts by joining those that are closest together and then keeps joining. So we start with everything spread apart and we join them gradually until we build the tree. So we're going to start with the list of trees that only have one element each, and then we're going to join them together and reduce the number of elements in this list, the number of trees in this list, until we have everything in a single list. To do that, we're going to use the same function that we have created last week, and I'm going to assume it's in this file. If you have a different name, just import whatever module you created for the code for last week, but it's this function, combine element. So basically we receive a list, two indexes and the new element, and what this does is it creates a new list where the first element is replaced, the first index here is replaced by the new element, and the second is discarded. So basically this is what we used to combine elements during the step-by-step procedure in UPGMA. So this is a function that we already have from last week, and we're going to use it now to join two branches. This would be the list of trees that we have. Remember that UPGMA starts with everything spread apart. It starts joining them together in small trees and then larger trees and so on. So this would be the list with all these small trees. And this is the matrix with the distances. So from the module that we had last week, we're going to call the UPGMA step function, the one that checks which indexes correspond to the smallest distance and computes the new matrix. So this is what we're going to get here, the two indexes and the new matrix. We're going to do the same thing as before. We're going to split the distance between the two of them, between these two indexes. We're going to split it in half so that each sub-branch gets half the distance between the offspring. And we're going to join these two nodes. So the way we're going to join these two trees here is to create a new dictionary, dictionary that is the start of a tree. We're going to put each of these nodes in the subtrees of this dictionary. So one is to the left, the other to the right. And now we're going to fill the rest of the information we need for these nodes. So the total length so far now is the distance between these two in the matrix. This is the total length that we need to go from here, this particular node, all the way to the end. So each dictionary here is representing one branch of the tree. If we go back to this diagram, for example, if we want to join this subtree with this subtree here, we need to create this dictionary and assign left to this one and right to this one, for example. And now we have this dictionary representing or giving us the starting point for this whole tree. And this is what we're going to do here. We're going to put the total length, which is the distance that we need to go all the way to the end of the tree, to the leaves. We're going to create this entry support with a 0 because we did not find support yet. We did not test the different replicas. And we're going to create this elements list. So to do this, we're going to add the list of elements that we have in one of the nodes to the list of elements in the other node. And then we're going to sort everything. So this sorted function returns a list with all of these elements sorted. And the reason we're going to do this is that now it's very simple to check which species are falling below this branch, so in this subtree here. And now we're going to update the list of trees by discarding the second node, that one. We're going to take it out of the list. We don't lose it because it's here attached to this new tree. And we're going to replace the node 1, which is the one that we stored here on the left. We're going to replace it with this new node. So the list of trees is going to reduce by one element, and the first one is going to be replaced by this one here. So this, what it does is, if we have, for example, these two trees in our list, the one with Homo sapiens, Pan, and these syndactyls, and then all the elephant and so on. If we have these two subtrees in the list, we're going to throw them away from the list and replace them with a single subtree, which is this one here that we created now. Okay, so this is one step in UPGMA where we follow the UPGMA algorithm, look at the smallest distance, join those trees together. Now we're going to do this until we only have one tree. So we're going to create the original differences matrix. Note that this is also in the module that we created last week. We're going to create an initial list of trees where each tree is just one node containing one element, and we store here the index of that element. There is no subtree because this first list of trees is only storing these final nodes here, Homo sapiens, Pan troglodytes, and so on. So we don't have anything under those nodes. And the elements is only a list of one single element, that particular one. And now, while the number of trees that we have is greater than 1, we're going to keep calling this join branches function to select the closest ones, combine them together, and return the new matrix, which is now smaller, and the new list of trees, which has one fewer elements. So we're going to repeat this until we have everything gathered together in a single tree, and then we return that tree. So this 0 is because the trees variable is a list of trees, and at the end, when we only have one tree, tree 0 is the only tree left that we have. So what is a tree? A tree is at the head one dictionary that looks somewhat like this, but then each dictionary has a left and a right sub-branch that we can use to follow along the tree. Okay, so now we're going to need to create replicas of the multiple sequence alignment. For each replica, we can create a new tree. We already have this here. We have the bootstrap to create the replica. We have these functions to create the tree. But now, for each node in the original tree, we have to check if there is a branch present in the replica with the same elements. This will indicate there is support for that node, for that branch in the replica. So in order to check if a branch is present in a tree, we're going to need to use a recursive function because the tree has each node of the tree has this left and right subtrees, and we need to follow them both to seek for the right node. So if we receive here a tree can be the large tree or can be any subtree, any branch, and a list of elements sorted, the sorted list of elements, we can check if this branch corresponds to grouping these elements together by comparing the elements entry in our tree dictionary with this list of elements. So if it's the same, it means that this branch here, this tree, contains exactly grouped together these elements. So in this case, we return true. Now, if this does not happen, we're going to check if the length of the elements entry in this tree is larger or the same or smaller than the length of the elements that we are looking for. Because if this is the case, then we cannot find these elements in this group in any subtree of this tree, because we already have only the same number of elements at this branch, and below the branch we will only have branches with fewer elements because we keep splitting them in half and so on. So in this case, we are going to return false. Or if, for example, the left branch of the tree is none. The way we're creating the tree, if the left branch is none, then the right branch is none too. We don't need to check both of them. But this means that we reach the leaf of the tree. And in that case, if we did not find the elements there, we're going to return false. So these are the terminal conditions. If we find the elements, we return true. If we know that we cannot find the elements in any subtree starting from this tree, we return false. Otherwise, we need to check. So we're going to return whatever comes as the result of checking for the left subtree for these elements. So this can be true or false. If this is true, then we return true. Okay, we found the elements somewhere here. Somewhere in this tree, there is a branch that contains exactly these elements. If this is false, then we're going to check the right subtree to see if there is a branch that contains exactly these elements. So this is the recursive function that we can use to iterate through all the nodes in the tree, looking for any branch that contains exactly the elements that we have here. And now we're going to use this to update the support in our main tree. So this is the main tree, this is the replica, and the main tree can be any, it can be the initial branch or any sub-branch, because now we're going to check if the length of the number of elements on this subtree is greater than 1. Because if it's only 1, we don't care. We don't care for support for those nodes that are already the terminal nodes, because we know those appear in all trees. They are just the individual species. It's only the groups that we care about. So if this particular node, if this particular branch has more than one element, we're going to check if this combination of elements here is present in the replica tree. If it is, then we're going to update the support of this branch and add 1 to it. So we're going to increment. Remember again this plus equal operator here, so that we keep incrementing these values. And then we're going to do the same thing for the 2 subtrees. So we start at the top of the tree, and we're going to repeat for each branch of this particular tree here, and each of this branch will be a smaller tree, and we're going down along the tree until we reach the leaves. Okay, so now the only thing that we need to do before putting this everything together is to create the text in Newick format so that we can see the result at the end. Now, in last week we did not create the tree, we just created the Newick description of the tree, so we did not need to use these more elaborate data structures. Now we have the tree created as this chain of dictionaries and dictionaries in the left and right branches and so on. So now we're going to create the tree, the Newick description from this tree structure that we created. So what we need is the list of names, because what we are storing in our tree is the index of each species in the list of names. So now, in order to write the names to the Newick format, we need the list with the names, and we need the tree. So one thing that we can check is if there is no more tree, right or left, it doesn't matter. We could do it like this. It will be the same, because when we reach a leaf node, neither both right and left are none. We don't have any more entries. Or we could also do this, for example, if if the length of the elements in the tree is equal to 1. So there is only one element in this particular node. So this would be an alternative to check here. So this is the terminal condition. When we reach a leaf, and a leaf is something that has no subtrees or only has one species, then we just return the name of that species. Otherwise, what we're going to do is we're going to get the left and right text from the subtrees. So the left it will be the Newick representation of the left tree that we have here, starting from this one, so the left branch, and then added to that the length that we need to cover so far, which would be the total length at the current branch that we have minus the length of the total length of the left branch that we are putting here. So this would be the entry for the left subtree. Note this this column separator here that is separating these two. So the text of the tree and the length of the branch there. And we are going to do exactly the same thing for the right sub-branch. So this is the same notation that we saw last week for the NAWIC format, where we have the branches and after a column a value so that we can represent the length of those branches. But we also need to include the support. And we're going to use an extension to the NAWIC format that includes the support in this value here before the column marker indicating the length. So this is the notation here. We have the branching, and here we have the support, and then a length, and then here another support and the length. So what we're going to do here is we're going to create these branches here with the parentheses. We are going to put the left and right text, and then we're going to add the support there by looking at the support field in the current tree that we have. We are not going to put support on the leaf nodes because this does not make sense, but we're going to put support every time we have these parentheses and keep moving up the tree. So now we're going to put everything together. We're going to load the cytochromes sequences. Remember that this load fast is a function we created last week, so it's in the multiple sequence alignment module. We're going to get the species names from the information on these sequences. So this is also on the module from last week, and we're going to compute the alignment. And this is the same as we did last week. So this is basically what we did last week. We're just using the functions on the multi-sequence alignment module. Now we're going to create the tree, and this create tree is the new function that actually creates this representation of the tree using those dictionaries, that data structure that we saw. And we're going to repeat 100 times, creating a bootstrap replica of the original alignment that we got here, and then creating the tree for that alignment, for that multiple sequence alignment, and update the support of our original tree by checking which nodes are present on the replica tree. So which groups of species in the replica tree are the same as in our original tree. And now we create the NAWIC text and we can visualize the result. So this is what you probably will get different numbers each time you run, but they should be similar to this. So one thing that you can see is that this group of 3 species, this is a lemur, I think, chimpanzee and humans, they always are together in a clade. The human chimpanzee grouping only appears 50% of the time. And the reason for this is that these are all equally distant. So in some replicas, we get grouped with the lemurs. In others, the chimps get grouped with the lemurs and so on. So this is not, from the cytochrome sequences, we cannot be too confident on this branch, but we can be very confident of this branch. These always come together. These two also are together most of the time, but these grouping all of these exactly like this, so all of these in the same group with nobody missing and no added species, only happens 60% of the time. Probably because sometimes we get a different grouping, one of these falls out or something like that, and then we have this branch separating these two sub-branches. This is also occurs very often in the replicas and so forth. So basically, by looking at how often the particular groupings of species appear in our replica trees, we have an idea of how confident we can what confidence we can have on each particular branching. So to sum up, we saw this bootstrapping as a method to create replicas by resampling at random from the actual data that we have. We cannot typically resample from the population, but we can resample from the sample that we have, and that's why bootstrapping is very useful. This is basic bootstrapping is good when the values are all sampled at random, so we can just generate new examples at random. For example, we can assume that the multi-sequence alignment columns are independent, and we can just generate new multiple sequence alignments by selecting those columns. If we have a situation where some of the values are actually chosen or fixed by us, for example, substrate concentration in the Michaelis-Menten kinetics example, then we cannot use basic bootstrapping, but we can use residual bootstrapping, or there are many variants like wild bootstrapping and so on, but the idea is basically the same. We are resampling some aspect of our data in order to create replicas and then compute statistics from those. So for this week, the main exercise will be computing these three with support, and so you can review this step by step and see how it works by actually implementing the code.